Hi, welcome to my little video here. We're going to take a look at bacteria and viruses, and then I'm going to introduce to you the Gram-Stain lab that we're doing in class coming up. So I just want to highlight some of the main ideas here. Bacteria are more common and more influential to our lives than archaea. If you think back to Unit 1, you'll recall that archaeans are found mainly in extreme environments, like frozen places and boiling hot springs, thermal vents at the bottom of the ocean, where you and I don't spend a whole lot of time. And that's why bacteria are um, really more a part of the human life, is because they can be found in the places that we live and even inside our bodies. They have harmful and helpful roles. And this picture here will give you a little idea of some of the various shapes that bacteria cells can have. So though they're too small to see, when you look at them through a microscope, you can describe what you're seeing by using terms for these various shapes. There are lots of structures that are found in most or all bacteria cells. The things that all cells have: DNA, cytoplasm, ribosomes, and a cell membrane. They also have, every bacteria has a cell wall. Many have flagellum for movement. They have a chromosome, which is not in a nucleus, but is in the cell. And they may have pili, they may have a capsule, and they may have an endospore, and things like that. Notably absent, there's no nucleus, there are no membrane-covered organelles. There cannot be a chloroplast or mitochondria or anything like that inside a bacteria cell because they are prokaryotes. Just a few examples of helpful bacteria. I'm going to go into detail on two of these. In the human body, in our digestive system, we are not able to digest all of the things that we eat. So some of the things that we eat are digested by bacteria living inside our system. So lactobacillus is an example of a helpful gut bacteria. E. coli is also normally and naturally found in our lower intestine and can have a helpful role there. Nitrogen fixation and environmental decomposition are another one I wanted to pinpoint for you. In the soil, if there was not bacteria in there, if there were not the types of bacteria that you do find, you would have problems like dead things would just sit there. The leaves that fall in the fall would not go anywhere. They would just stay there. And plants wouldn't be able to get nitrogen to grow. So decomposition and changing the form of nitrogen into a form that plants can absorb in their roots and use are two really important bacterial roles in the environment. Last but not least, different aquatic species that light up, like fish and squids deep in the ocean, are not actually glowing. What they're doing is keeping a pocket of a bacteria called Vibrio fischeri inside them. And the Vibrio are glowing. So it's actually bacteria that have bioluminescence and not these fish and squids. They just host a colony in their body, and that's what's providing the glow, that light. Okay, now of course bacteria can be harmful as well. Some common infections that can be caused by bacteria: pneumonia by pneumococcus bacteria, pneumococcus, staph infections and blood poisoning. A lot of infections that people get after surgery in a hospital or get infected in wounds can be staphylococcus. Food poisoning, even E. coli, which is normal to live in the lower part of your digestive system, will make you very sick if it gets into food and enters your mouth in the upper part of your digestive system. Salmonella and Listeria are other bacteria that can contaminate foods and make us sick when we consume the foods. Okay, tuberculosis and syphilis are other very serious infections that can be caused by bacteria, and there are many more. This slide is comparing viruses to true cell-based living things like bacteria all the way on up to animals. Where all bacteria and all cell-based life have a genetic code that's made of DNA. Some viruses have DNA, but others have RNA. You could see which is which on the next slide. Where there's a lipid bilayer around every cell, and there are ribosomes in cells to make essential proteins. Most viruses don't have a lipid bilayer. Some of them do, but they borrow it from cells that they're reproducing in. And viruses don't have ribosomes. Bacteria and other living things can be free living, or they may require another organism to complete part of their life cycle. Viruses always require an organism to complete their life cycle. They have to go inside a cell to use the ribosomes to make their proteins. And some of them, when they escape, take some lipid bilayer with them for a membrane. Okay. Like I said, many viruses, such as the coronaviruses and a lot of their relatives, are made of RNA genetic material in a protein coat. Others are made of DNA, shown here in blue. The last thing I want to make sure you know about viruses is that they can reproduce right away. So they enter a cell, they immediately take over the cell, use the ribosomes to make new proteins, copy their genetic material, and assemble new viruses, bursting out of the cell and killing it, which alerts the immune system, if you're in an organism like a human, and causes a response. The lysogenic cycle is a little bit sneakier. The virus gets into a cell, and instead of reproducing its own parts and breaking out of the cell, the DNA, or a DNA version of the RNA, from that virus goes and sits inside your chromosomes. And then, as the cell divides, every time the cell divides, it copies its DNA, copies the viral DNA with it. Eventually, one day, the virus has had enough, it pops out and starts doing the lytic cycle, where it reproduces copies of itself and bursts out. So if a virus is using the lysogenic cycle, it can actually be inside a living thing without causing it any noticeable damage for a long time, and then suddenly have its effects. You'll find some more information here about the immune response that humans can have to viruses like the coronavirus. I put it here because I think it's interesting for you. I'd be happy to take questions about it, but I'm not going to go into detail in this video. Okay, turning our attention back to bacteria. One way to tell different bacteria apart is by looking at their shapes, like I mentioned before. You can also use a test called Gram staining to stain the bacteria different colors based on its cell wall. Every bacteria has peptidoglycan in its cell wall, but bacteria with more peptidoglycan in their cell wall stain a dark purple color as a result of this process, while cells with a thinner peptidoglycan layer will end up turning a reddish pink as the result of these steps. Okay, the lab will explain everything as you go along. I just want to make sure you know how to find your way around the lab. So you're going to click begin, or since I already have it open, I'm going to click continue. And it's going to narrate for you. Once it reads what's on screen, you can get some more info here or continue to the next step. This menu here will take you to the different chapters, which are each reflected in the document where you're taking your information. Okay, so as you continue through, you'll have the opportunity to read more about different things, but the questions that I ask you will be from the main statement. As we move through, there will be steps where it asks you to complete things on your own. So it'll ask you to do different motions. A large arrow will come up and tell you how to move. So clicking and moving different items around will help you complete the Gram stain. Okay, we'll be doing this in class, so you'll have support from me as you need, but I wanted to just give you a little familiarity with how to move around it. If you were to stop and have to finish on your own time, again, you'd come to this menu to go to whichever step you needed to resume. Okay, I think you'll really enjoy this lab. It's very grounded in reality. You're making a decision about yogurt. So it's actually a, you know, business purpose that does use gram stains in real life. And each step is explained as you do it. You're going to just follow along. You're going to answer the questions in the document as you go and hopefully enjoy it. By the end, you get to try one on your own and see if you can do it without any mistakes. All right. I'll see you in class. Have a wonderful day. Bye now.